So for this study, what we did is we looked at the cell-free DNA fragmentomic signatures in the peripheral blood. Originally, we tried to do this as an MRD test. The idea being, let's say that we don't have any baseline sample from a patient. We don't know the mutations to look for in the cell-free DNA. Can we just look at the characteristics of the DNA itself to predict MRD status? And the reason we tried to do this is because the cell-free DNA is not random, where it's cut, where we see more of it, where we see less of it in the peripheral blood. So what we did was we looked at genes that are likely to be overexpressed in multiple myeloma using ATAC-seq. And we looked at the coverage of transcription start sites of those genes in cell-free DNA to predict if the chromatin was open, so that gene was being transcribed, the DNA would be unprotected, and we'd see less of it at the transcription start site. And also the fragment length, with the idea being that cancers tend to have more active transcription in general, more open chromatin, which leads to shorter cell-free DNA fragments in the peripheral blood. Now, using fragmentomics alone, we were unable to get very high correlation with bone marrow-based tests for MRD status because we didn't have the sensitivity. Now, I think that there's future work that could potentially get us closer to there, which is in development. But what's really interesting is we then took those signatures and the cutoffs that we identified at the MRD time points in 41 patients, MRD positive and negative, and we applied them at baseline, at diagnosis, in 43 patients to see what that looked like. And we found that most patients showed this MRD positive signature at diagnosis, which is what we'd expect. But there was a subgroup of patients that showed MRD negative cell-free DNA characteristics even at baseline. And what we found was the patients that were MRD positive tended to have earlier progression events, whereas those that were MRD negative tended to respond well to treatment and remain in progression-free for long periods. Further, the patients that were MRD positive by the cell-free DNA fragmentomics tended to have more high-risk cytogenetic features in myeloma compared to the MRD negative groups. So what we think is that patients that have more cell-free DNA positive or circulating tumor DNA positive signatures at diagnosis may be higher risk patients that tend to progress earlier at diagnosis. And this could be another biomarker that we check when we're evaluating patients to understand their risk of a progression event and response to therapy. Now, this again was only around 43 patients that we tested in, so we need to evaluate this in larger, bigger cohorts. But we hope that this technology of cell-free DNA fragmentomics can be another tool that we can use from the peripheral blood to identify patients at risk of early progression or those who are likely to respond well to treatment. And this might be especially useful in cases where we have limited bone marrow specimen, for example, to perform standard cytogenetics for risk stratification.